Today we'll take up the portion that is antigen processing and presentation. All the time I kept mentioning in the immune response, this process is very important, and without the processing of antigen, immune response will not take place. And I had also mentioned that antigen processing takes place through the antigen processing cells. There are special cells meant for this, which process the antigen and present it on class 1 and class 2 MHC. Now let us see which are the cells that are involved in antigen processing and presentation. First, the professional antigen presenting cells. Means they're only processing of the antigen and presentation. Of course, there is B lymphocyte, which does both production of antibodies. At the same time, it also has the ability to process the antigen. Now, professional antigen presenting cells are dendritic cells, macrophages, and B lymphocytes. I had mentioned clearly earlier, it is only the antigen presenting cells have the ability to express both class of MHC, that is class 1 and class 2 MHC. No other cell has this ability. Only antigen presenting cells can express both these classes, class 1 and class 2. Now, what is the function of class 1 MHC? I have already explained to you that it is meant for presenting intracellular antigens of the cellular origin. That includes the antigens of the pathogenic origin, that is bacteria and virus. And I already mentioned that class 1 MHC molecule is the fingerprint molecule that identifies a particular tissue, because the class 1 MHC that is expressed by liver cells will have the antigens characteristic of liver cells. MHC expressed by the kidney will have characteristic of antigens expressed by the kidney. From this, you can identify to which tissue that class 1 MHC belongs based on the antigens expressed by class 1 MHC. Generally, the function of class 1 MHC is to express the antigens of the tissue origin or the cell origin of that particular tissue. But that cells can get infected by virus or bacteria. In such a condition, the class 1 MHC by default also will express the antigens of bacterial or viral origin if it is infected. All throughout, it will be expressing self antigens. But by accident, they can express non-self antigens, that is of bacteria and viral origin. Then the immune response gets triggered. Okay. So, class 2 MHC molecules, which will be expressed only on antigen presenting cells. They are not going to be expressed by the other cells. But antigen presenting cells also have class 1 MHC. Simultaneously, they can express class 2 MHC. That is the characteristic of antigen presenting cells. And class 2 MHC is meant only for presentation of antigens of exogenous origin. They are all foreign antigens, may be derived from plants or animal origin, or they can be bacterial or viral origin. Exogenous antigens, they do not belong to our body. They enter our body and then they are processed by the antigen presenting cells, and then they are presented on class 2 MHC. Now, how these processes will take place in the antigen presenting cell, we will see. That is presentation of antigen on class 1 MHC of endogenous origin, presentation of antigen on class 2 MHC of exogenous origin. So, the non-professional antigen presenting cells, skin fibroblast, glial cells in the nervous system, pancreatic beta cells, thymic epithelial cells. We have seen how important they are in positive selection of the T lymphocytes. That is thymic epithelial cells, thyroid epithelial cells, and vascular endothelial cells. All these are non-professional antigen presenting cells, called as non-professional because they actually are not meant for antigen presentation, but when demanded, they will involve in antigen presentation. They will gain the characteristic of antigen presentation and they will present the antigen, but they are not professional. Now let us see, we have seen the structure of class 1 MHC, and I hope you remember it has got an alpha chain made up of alpha 1, alpha 2, alpha 3 and beta 2 microglobulin, and the antigen is presented in this antigen groove made up of alpha 1 and alpha 2 domain. Okay, and that process of presentation of antigen on class 1 MHC is called as cytosolic pathway because it originates in the cytosol. The antigen is present in the cytoplasm and that antigen is processed and presented. Here it is mentioned as viral protein. It could be on bacterial protein or it could be in even the cellular protein. Okay, if it is cellular protein, it is self. If it is bacterial and viral protein, it is non-self. All proteins that are there in the cytoplasm, once they finish their function, they are supposed to be degraded and broken down into bits and pieces of micropeptides, and finally those micropeptides will enter the endoplasmic reticulum, which is shown here. This is the endoplasmic reticulum, lumen of the endoplasmic reticulum, and there they further undergo breakdown and produce amino acids, and those amino acids will be utilized on the surface of the rough endoplasmic reticulum by the ribosomes when the protein synthesis is taking place. So you know now protein turnover, the proteins have finished their lifespan and work will be broken down. What is the apparatus that breaks down the protein? That is called as proteasome. Proteasome. Please note the spelling. The proteasome is nothing but a huge pipe-like structure. Huge in the sense it is not visible to the eye, but it is quite a macroscopic structure which is visible in the cells which are active in antigen processing, and that tubular structure has in the inside surface all proteases lined up. It's a lineup of proteases formed into a tube with ATP attached there. ATP will be utilized as an energy source for breaking down the proteins. The proteins that are supposed to be broken down will enter from one end, and then broken down proteins will leave the other end. And that is the function of proteasome. It's a body meant for breaking down the proteins, and generally in the cell, the proteins that are supposed to be broken down are ubiquitin labeled, ubiquitin attached. They are also called as ubiquitinated proteins, and ubiquitinated proteins are supposed to be broken down. Means they have finished their work. And then the broken down protein in the proteasome is transported across the rough endoplasmic reticulum membrane through a protein channel called as TAP. Okay, the TAP. The TAP has the structure like this. And what is the full form of TAP? Transporter associated antigen processing protein. Transporter associated antigen processing protein. It is made up of two of proteins, glycoproteins, attached to the membrane of the rough endoplasmic reticulum with ATP within themselves and a channel. Through that channel, the broken down protein traverses from the cytoplasm into the lumen of the rough endoplasmic reticulum. That is what is shown here, TAP. But please remember, the detailed structure of TAP is not necessary to be given in the examination. You are expected to give this diagram for cytosolic pathway of antigen processing and presentation or cytosolic pathway of endogenous antigen presentation. Now let us say that the broken down protein enters through the TAP into the lumen of the rough endoplasmic reticulum. In the rough endoplasmic reticulum, further that micropeptides are broken down. And at the same time, the rough endoplasmic reticulum is also synthesizing the class 1 MHC molecule. That is the alpha chain of the class 1 MHC molecule and the beta microglobulin molecule is synthesized independently. And actually that protein is produced outside on the ribosome. And once that protein is produced of the alpha chain and beta 2 microglobulin, it is internalized into the lumen of the rough endoplasmic reticulum. And after internalization, the class 1 MHC molecule assembly should take place. Meantime, remember that the proteins that are broken down have come into the rough endoplasmic reticulum. They are also getting broken down. The internalized class 1 MHC molecule should not be broken down. It should be protected from breaking down. And that is done by a molecule called as calnexin. CALNEXIN, calnexin. The calnexin molecule comes and sits adjacent to the internalized class 1 MHC molecule, okay, which is not yet joined with the beta 2 microglobulin. It is partially formed with alpha chain. It goes and protects it so that it is not digested. Okay. Then meantime, there are other molecules like, calnexin has come and protected it. There are other molecules now. As soon as the beta 2 microglobulin comes and joins, class 1 MHC is complete. Class 1 MHC is complete. And it has gained the structure, that antigenic groove, where the micropeptides come and lodge. They can sit there. But remember, class 1 MHC can accommodate or make the peptides to come and bind to that antigen cleft if they are only of 10 amino acid residues. If they are more than that, it cannot accommodate. Okay, so only about 10 maximum. So such peptides can come and bind there. So until then, until that peptides, which are broken down molecules of the antigen, come and bind there, that molecule still needs to be protected, which is fully formed now by joining the beta 2 microglobulin. And that is done by two more molecules called as tapacin and calreticulin. Calreticulin and tapacin also come and bind there. See here, tapacin and calreticulin, which have come and bound to the fully formed class 1 MHC molecule. By then, calnexin has finished his work. It has left. So what are these molecules? Tapacin, calreticulin, and calnexin are called molecular chaperones. They are called as molecular chaperones, means they are protectors and helpers of the class 1 MHC. Now the class 1 MHC, which is protected by tapacin and calreticulin, should bind to the antigen in its antigen groove, okay, from the antigens that are generated here, micropeptides that are generated here in the lumen of the rough endoplasmic reticulum. Those could be self proteins, or those could be the proteins of viral origin or bacterial origin. So if the self protein is presented here, okay, that class 1 MHC molecule now emerges out of the rough endoplasmic reticulum by blebbing called as bubble. It will be a vacuole-like structure, bubble, in which the class 1 molecule bound to the peptide is internalized. And that bleb or the vacuole, which is enclosed, having class 1 MHC, migrates to the Golgi complex. It goes and fuses with the Golgi complex. In the Golgi complex, once again, the class 1 MHC from there emerges as a new bubble, But now that bubble that has emerged out, which is a vacuole containing class 1 MHC, will have the membrane around it exactly similar to the plasma membrane. The chemical composition of that bleb will be similar, or vesicle, you can call it as a vesicle, not vacuole, vesicle, similar to the membrane structure of the plasma membrane. So from there, from the Golgi complex, it emerges out and migrates towards the plasma membrane and fuses with the plasma membrane from inside and everts out, inside out, everting out. When it everts out, the class 1 MHC molecule is expressed on the plasma membrane with the antigen bound to its antigen groove. Now you realize here, if it was a self antigen of the cellular origin, which was processed inside the lumen of the rough endoplasmic reticulum, which had bound to the class 1 MHC antigen groove, that will facilitate the class 1 MHC to present the self antigen. And the presentation of self antigen will not trigger immune response. In the antigen presenting cell, this process is a normal process. But then, if the antigen presenting cell or the cell of a particular tissue, if it is infected with a virus or bacteria, the viral or bacterial protein will also go through the same process of processing, entering through the tap after proteasome breakdown, entering through the tap into the lumen of the rough endoplasmic reticulum, assembly of class 1 MHC molecule that is taking place there, and by accident that viral or bacterial protein of 10 amino acid residues will come and lodge itself on the class 1 MHC antigen group, and it will be transported across the cytoplasm in this manner, first from the rough endoplasmic reticulum as a vesicle, and that vesicle comes and fuses the Golgi complex. Then it will emerge as a vesicle, but the membrane of that will be almost similar to the plasma membrane. That facilitates that vesicle to fuse with the plasma membrane from inside surface so that it can evert out. And when that class 1 MHC now, when it presents that viral peptide or bacterial peptide, that is going to be recognized by the immune cells as a non-self protein. If it is presented by the antigen presenting cell, okay, well and good. It is going to be presented to CD8 T cells, which recognize the antigen presented on class 1 MHC, and they are restricted to class 1 MHC, and they get activated. Or it could be presented to NK cells. NK cells also recognize the antigen presented on class 1 MHC. That too, self antigen of viral or bacterial origin. So the intracellular pathogen that is there within the cell is now going to be recognized by its antigen being presented on class 1 MHC. And once that is recognized by CD8 or NK cells, you know what they do. They get attached to that particular cell. If it is the host cell and not an antigen presenting cell, normal tissue cell, then also the antigen that is presented on class 1 MHC is recognized by CD8 and NK cell. And they get arrested there, they get activated, they will start secreting granzymes, and they will start secreting the perforins, which are inserted, and that cell is going to be destroyed. We have seen that process in the effector mechanism. Okay, so the antigens of endogenous origin, that is of cellular origin, are presented on, and if the cell is infected by virus or bacteria, which is an intracellular pathogen, those antigens are also present on class 1 MHC. Now, this process of transport of antigenic peptide bound to class 1 MHC across the cytoplasm to the membrane is blocked by some viruses. There are viruses like HIV virus, herpes virus, okay, HIV, herpes virus, and there are several other RNA viruses which can block this process of presentation of antigen on class 1 MHC on the surface of that particular cell. Then that cell will not be recognized for the foreign antigen. But you know that I have told you, if class 1 MHC expression is failed, cells have the special ability to recognize that particular cell which is not expressing class 1 MHC and then attack that cell. Okay, so cytosolic pathway of antigen processing and presentation involves presentation on class 1 MHC. What is the ultimate mechanism? The class 1 MHC is synthesized within the rough endoplasmic reticulum. In the sense, it is assembled. Protein of that is synthesized outside, that is in the ribosome. It is assembled within the rough endoplasmic reticulum lumen. It is protected by calnexin, molecular chaperone, until beta-2 microglobulin comes and joins it. Once beta-2 microglobulin joins it, the molecule is complete. As soon as the molecule is complete, it is joined by two more chaperones, that is stapacin and calreticulin. By then, the calnexin will, and now that molecule, class 1 MHC molecule, should receive the antigenic peptide in its groove. And once it receives the antigenic peptide in its groove, it has to leave the lumen of the rough endoplasmic reticulum by forming a vesicle. By then, those two chaperones which were protecting it will leave it alone, that is stapacin and calreticulin. From there, it exits rough endoplasmic reticulum and migrates to the Golgi complex and then forms a vesicle and migrates to the plasma membrane and fuses with it from the internal side, from the inside. Then we will see class 2 MHC, that is processing and presentation on class 2 MHC. To recall your memory back, the class 2 MHC differs in structure from class 1 because it is made up of two different chains, that is alpha and beta chains. Both are anchored on the plasma membrane through a transmembrane domain plus cytoplasmic domain. The antigenic groove is made up of alpha 1 and beta 2 domain where the antigen binds. Okay, now how does this happen? We will see. So the ability of processing the exogenous antigen is present only in the antigen presenting cells. Normal tissue cells will not do this function. So therefore, expression of class 2 MHC is unique only to antigen presenting cells. And it is not expressed by normal other nucleated cells. Okay, it is presented by the antigen presenting cell. Let us take the exogenous antigen. Now, this exogenous antigen, as I mentioned, it could be of plant origin or animal origin or tissue origin of other individual, like when the tissue or organ is donated or tissue graft is carried out in a person, the antigens of the other person will be different from the antigen of the person who is receiving the graft. So the graft tissue that is donated will have some different antigens, and those antigens become exogenous antigens. Then antigens of bacterial origin, antigens of viral origin. Okay, they are present, maybe circulating in the body fluid. Okay, and they are going to be now processed by the antigen presenting cell. So there are different mechanisms of processing. One, if it is a soluble antigen, it will be pinocytosed. If it is a solid antigen, it is going to be phagocytosed. And there is also possibility the molecular antigens like proteins, glycoproteins, lipoproteins, all these types of molecular antigens may attach to the receptor of the cell. They have receptor. And remember, I had mentioned those receptors. Pathogen associated molecular patterns. In that, I had explained to you those receptors which are called as toll-like receptors. Toll-like receptors, TLRs. I told you about them. Okay. Pathogen recognition receptors, PRRs. And those receptors will bind to that molecular antigen and antigen in. If it is receptor mediated intake, it is called as endocytosis. If it is solid particle intake, it is called as phagocytosis. If it is liquid materials, it is soluble material, then it is pinocytosis. Whatever may be the case, once it is internalized, that endosome or phagosome or the pinosome is supposed to migrate inside and fuse with the lysosome to form phagolysosome, okay, and get digested. And whatever that antigen that has been internalized is going to be digested in the lysosome. Now let us see simultaneously what is happening inside in that particular cell. Only antigen presenting cell. In the antigen presenting cell, in the rough endoplasmic reticulum, the class II MHC is getting assembled. It is synthesized outside on the ribosome as alpha chain and beta chain. Okay, alpha and beta chain. After the synthesis of that alpha and beta chain, it is internalized. And when it is internalized, just like the way class 1 MHC molecule was protected from digestion, this is also going to be protected from digestion. Class 2 MHC, which is internalized, associates with a protein called as invariant chain, which is also a molecular chaperone. There, in the case of class 1, it was calnexin. Here, it is invariant chain. That invariant chain comes and lodges itself on the antigen groove. Here, antigen groove made up of alpha 1 and beta 1, sorry, on the surface, on that groove, it will come and lodge. It will bind there. And you know that the class 2 MHC can bind to the peptides of length of 13 to 18 amino acid length. And the chain overhangs on either side. Okay. And the chain comes and binds there. And now once the chain is bound, invariant chain is bound, that molecule is protected. The completely formed class 2 MHC bound with invariant chain now emerges out as a vesicle. And that vesicle bound inside with class 2 MHC bound to invariant chain goes and fuses with Golgi complex. Same pattern. Once it fuses with the Golgi complex, it will form another vesicle and emerges out as another vesicle. And that vesicle which emerges out from the Golgi complex by chance now can fuse with a lysosome. And when it fuses with the lysosome, that invariant chain protein that was bound to it is going to be trimmed. It is going to be trimmed. All that excess amino acids which were there on either side are going to be trimmed and formed into a clip protein, CLIP, clip protein. Okay, and clip protein, you can expand that also as class 2 associated, okay, class 2 associated chain protein, clip protein is expanded as class 2 associated invariant chain protein. And that invariant chain protein is trimmed, and the trimmed protein is bound as clip protein. At the same time, that lysosome can also fuse with the endosome or the phagosome or pinosome that was formed with the exogenous antigen. And when it fuses with that, you have now class 2 MHC, which is fused with the endolysosome, and it is there in the internal surface with the clip protein. And remember that endosome or phagosome or pinosome was formed from the exterior membrane of that particular antigen presenting cell. And that plasma membrane of the antigen presenting cell will have a molecule on its surface called as HLA-DM. HLA-DM. HLA-DM stands for Human Lymphocytic Antigen Diversity Molecule. Human Lymphocytic Antigen Diversity Molecule. Diversity molecule means that molecule HLA-DM has diversity to bind to different types of proteins, just like the way class 2 MHC molecule has the ability. Okay. So that diversity HLA-DM molecule which is there on the membrane, which is also simultaneously internalized with the endosome or lysosome, okay, which is available facing inside in the endolysosome, okay, but its groove, HLA-DM groove, is empty. It is vacant. Let me show here HLA-DM, which resembles the class 2 MHC. Okay, it resembles class 2 MHC, but its groove is empty. All right. So the class 2 MHC has clip protein now. The invariant chain protein was protecting it. That invariant chain protein is digested, and now class 2 MHC is having clip protein. The clip protein containing class 2 MHC and HLA-DM are sitting side by side in the same vesicle. In the same vesicle. At the same time, in that same vesicle, lot of protein digestion is taking place because it is an endolysis, and the peptides that are generated, peptides are going to be generated, and those peptides generated are of the exogenous origin of the exogenous antigen. The HLA-DM which is there with the empty groove, like this empty groove, has strong affinity for clip protein. Since it is sitting side by side together with the class 2 MHC, and the class 2 MHC is bound with the clip protein, that HLA-DM will snatch that protein to itself because it has more affinity to it. The clip protein is snatched or robbed off, okay, from the groove of the class 2 MHC. The HLA-DM is a chain snatcher. It is a robber, chain snatcher. The clip protein is snatched from the groove of the class 2 MHC to the groove of the HLA-DM. So HLA-DM groove gets engaged with clip protein. So as soon as the groove of the class 2 MHC becomes empty, immediately that groove is going to be occupied by the proteins that were generated here, peptides that were generated here in the endolysosome by the digestion of exogenous antigen. So until then, the invariant chain is the self antigen, which was broken down to form clip protein. that was bound to the class 2 MHC, which was snatched away by the HLA-DM in the same vesicle because of its affinity. Now that antigen groove is empty, that is going to be attracting now the peptides of 13 to 18 molecules, 13 to 18 amino acid residues to its surface. So the antigen groove of the class 2 MHC is going to be bound by the now antigen of exogenous origin. That is, which is processed, which had come from outside, which had processed, and that processed antigen comes to the surface. That is through the antigen groove of the class 2 MHC. Together with the HLA-DM bound to the clip protein, now that vesicle migrates to the membrane. At present, the clip protein is bound to the HLA-DM, and the antigen groove of the class 2 MHC is occupied by the antigen of the exogenous origin. And on coming in contact with the plasma membrane from internal surface, once again it will fuse and evert out. By everting out, it will expose the class 2 MHC with the non-self antigen of the exogenous origin and HLA-DM of clip protein. Okay, HLA-DM of clip protein. And that is what is represented in this diagram here. So the HLA-DM will snatch the clip protein and make the groove available for the peptides that are generated from the lysosome by the digestion of exogenous antigen. So that is how the antigen cell processes the antigen of exogenous origin and presents it on the class 2 MHC. And antigens presented on class 2 MHC are going to be recognized by the Th cells. If the B lymphocyte processes the antigen like this, the B lymphocyte will present the antigen on class 2 MHC to Th cell, and the synapse will take place with the TCR, and beta 2 domain of the class 2 MHC will establish contact with the CD4 receptor, and that CD3 TCR complex will succeed in generating signal 1. So class 2 MHC restricts Th cells, and Th cells are class 2 MHC restricted. Without presentation of antigen on class 2 MHC, Th cells on their own cannot be activated. They have to be presented antigen on class 2 MHC. And class 1 MHC meant for activation of CD8 T cells and NK cells. So now you know the importance of both these molecules, class 1 MHC, MHC. And are classified under HLA, human lymphocytic antigens, because they were first detected and recognized on the lymphocytes. Even though the class 2 MHC is the characteristic of only antigen presenting cells, both class 1 and class 2 play a major role in immune recognition on self antigens. And without the presentation of antigens on class 1 and class 2 MHC, immune response cannot take place. You can understand the genetic deficiency where the genes for expression of class 1 and class 2 MHC are not present. And that syndrome is called as bare lymphocyte syndrome or naked lymphocyte syndrome, where it could be classified into two types. If class 1 MHC is not expressed, it is called as bare lymphocyte syndrome 1. If class 2 MHC is not expressed because of lack of genes, then it is called as bare lymphocyte syndrome 2. And there are individuals born like that. And if they are born like that, that also causes immunodeficiency. Please remember, they will have immune cells. But since class 2 MHC is not going to be expressed, if class 2 is not going to be expressed, TH cells are not going to be activated. If TH cells are not activated, they are major cells, T helper cells, responsible for both cell-mediated immune response and humoral immune response. Even CD8 T cells are dependent on TH cells. NK cells are dependent on TH cells. B lymphocytes are dependent on TH cells. So if TH cells are not there, if class 2 is not going to be expressed and non-self antigen is not going to be presented, there is no activation of TH cells. If there is no activation of TH cells, immune response fails. Okay. In class 1 MHC absence, if class 1 MHC is absent, then immune response will rely only upon those antigens of the viral or bacterial origin to be presented on class 2 MHC, and immune response will be triggered for humoral as well as cell mediated, but there will be no CD8 T cell response. There will be no NK cell response because they recognize antigen only on class 1 MHC. If class 1 MHC fails to present non-self antigen, CD8 T cells will not be activated, NK cells will not be activated. Therefore, there is a possibility that if those viruses or bacteria have the ability of inhibition of MHC, okay, and if class 1 MHC gene itself is not there, and those cells will be failed to be recognized by the NK and CD8 T cells, and those cells will not be killed. And such people who fail to express class 1 MHC will suffer from intracellular pathogenic infection. That is, viral and intracellular bacterial infection will be very, very predominant in those people. Remember, the persons who suffer from HIV infection, I mentioned that class 1 MHC is inhibited from expression in HIV infected patients. In addition, the HIV infected patients also will fail to show TH cell response, which we'll be talking about that in detail in Unit 4. Because of that, they suffer from immunodeficiency. And not only that,